Good morning, everyone. It's great to see you all. Thank you to the OIS folks for the opportunity to speak today and talk to you a little bit about what's going on with Stuart Therapeutics. We're developing a new drug class, collagen mimetic peptides, and these peptides have some pretty interesting characteristics. The mechanism of action, how it works, is the highly selective binding of our polypeptide, synthesized polypeptide, to disease-damaged collagen. That disease-damaged collagen repair results in a switch back towards homeostatic cell signaling, structural repair where collagen structures are involved, and a reduction in inflammatory activity in the affected region. One of the interesting byproducts of this is improvement in the cell microenvironment. We've demonstrated in our research studies improvement in epithelial cell environments, nerve cells, endothelial cells across the board. And I'm going to come back to the nerve cell impacts in just a few minutes when we talk about our dry eye program. We've completed a phase 2 study on our lead candidate, ST100, for dry eye disease. I'm going to talk about some of those results and what our findings have been about those results here in a moment. And we're about to complete our phase 3 trial. That phase 3 trial should finish up about the first week of January. We think that the use of CMP technology for dry eye offers an opportunity for a very comprehensive solution to the tissue damage issues that occur in the nerves, in the epithelium, and in the stromal tissues in dry eye. So in our phase 2 trial, we had quite a range of results, and they were very intriguing. We had visual acuity improvement with significance on day 2. We got symptom relief on a couple of different scores on day 14, staining scores on day 28, and unfortunately, because it was our first trial, we only measured Schirmer's responder rate and Schirmer's test on day 28, but we did hit significance on that day that we measured it. The drop was safe, very well tolerated. We had drop comfort scores that were consistent with popular artificial tear products. So we think the visual acuity improvements occurred due to early tear film impact. We're not certain of that, of course, because we didn't measure it, but we saw a very nice result on visual acuity right away in the trial. And there was a subset of patients that achieved between 2 and 3 lines of improvement in their visual acuity by day 14 versus 0 in the control group. From a symptom perspective, our significance was on the 4-symptom total score and on OSDI pain. We also saw very nice trends on burning, dryness, and grittiness in the results that we measured. Staining outcomes at day 28, total conjunctiva, total eye stain results were significant. And we think, again, based upon the totality of the results, the prolonged effect of nerve repair and inflammation reduction resulted in healing to the ocular surface. So again, tear production, we measured it on day 28. We got significant results in mean Schirmer's score as well as Schirmer's responder rate. Nearly 50% of the patients in the trial showed Schirmer's score improvements, so it was widespread amongst the patient population. And again, it's a very well-tolerated drop formulation. This is systematic improvement in the tear function without the use of an agonist. We used the control adverse environment chamber. I haven't shown you any significance results out of that. There are a few, but so far. But one thing that was notable about the drug, this is a chart that shows the cumulative change in ocular discomfort against baseline by visit day every 5 minutes in the control adverse environment chamber. And what this is showing us is that with continued use of ST100, the patients saw an improved ability to withstand exposure to dry air, high volume air, et cetera, irritants to the ocular surface. So there was a notable protective effect for the use of the drug. Now, we've talked, some other folks today have talked about the DEWS 3 workshop, the changes to the definition. As we looked at the totality of results and as we did additional animal studies, one of the things that jumped out at us was the nerve repair capability of the peptide. These images that you see on the screen are of a naive subbasal nerve plexus in the cornea of a mouse. Next to it is a dry eye model that uses atropine in a desiccation chamber to dry it out, damage the nerves, and you see the obliteration of the central portion of the nerve plexus there. And on the right, you see a treated version with ST100 repairing and restoring that subbasal plexus. We think that the corneal nerve repair and recovery explains the broad array of visual acuity symptom staining and tear production improvements that we saw in the phase 2 trial, and we hope to repeat in the phase 3, of course. So, vasocomatide, which is the drug substance in ST100, has the potential to be a groundbreaking dry eye therapeutic. We think it's a very comprehensive, broadly effective, and patient-friendly approach, and it solves a lot of the shortcomings that you see in dry eye therapies today. Speed of use is great, or speed of impact is very, very good for us. It's convenient, very comfortable, patient-friendly. We've got opportunity for all dry eye subtypes. We've got additional evidence that shows reduction in activated mast cells in lids in some preclinical studies as well. So not just inflammatory cytokines, but circulating inflammatory cells. From a commercial perspective, we've licensed the product to Aju Pharm in Korea for Korea and Southeast Asia. We retain the remainder of the worldwide rights ourselves, and we're looking for options to maximize the commercial value of ST100 once we're ready to go. We should have a drug approvable by late 2026. Now, we talked about bridging into the retina, so I took the opportunity, because we're a sponsor, to talk a little bit about what we're doing with this as we go into other areas of the eye. The areas in green are some priority areas for us. The other areas are potential additional indications for this peptide. Because collagen has prevalence throughout the eye and it's got a role in disease progression and chronicity, we think it's an ideal target for multiple indications that are going to be talked about at these sessions over yesterday and today. So as we look at myopia, I talked a little bit about the structural repair capability of the peptide. We've got quite a body of evidence, including an animal model that shows that we are able to restore the integrity of the posterior sclera, which is the tissue that is relaxed and causes the axial lengthening in myopia. In diabetic retinopathy, we just completed a series of studies that show repair of vasculature in the eye and in other tissues in the body. And we believe a good target for us may be anti-VEGF non-responders in diabetic retinopathy. And then finally, we've got a great partnership with the folks at Glaukos, where they've taken the lead on the development of this peptide in neuroprotection and glaucoma. Each of these areas has in vivo proof of concept data. We have some other tests that we want to run before we carry things forward, but the opportunity keeps expanding for this technology as we look at ophthalmology. You can really see the opportunity in one picture on this particular slide. This is a cross-section of a non-human primate outer retina and sclera. And one of the things that is notable about this is the fluorotag CMP is highlighted and binding and repairing collagen in multiple important regions of the eye. So you look at the internuclear layer, the synapse layers primarily. The bright green band is the interdigitation zone between the photoreceptors and the RPE cells. You see it in the basement membrane and in Brooks membrane, also in the choriocapillaris around the vessels and in the choroid, and then finally down at the bottom in the sclera. So this is what kind of lit the fire for us about the expansion of the opportunity in multiple areas. So we've got a new drug class. It addresses nerve, epithelial, and endothelial recovery in the eye. We get reduction in inflammatory markers and circulating inflammatory cells, and we get additional structural recovery for structural collagen in the eye. It's got a positive safety and tolerability profile, and we think the achievements in our dry eye program point to application in these other areas and de-risk the platform. I want to thank you all very much for the opportunity to talk to you today, and if you have questions, look me up. Thanks so much.